Hello, my name is Harsha Bukhari from the University of Pikeville, Kentucky College of Osteopathic Medicine, and I will be presenting my research I have so far on the vasodilatory effects of substance P. So, what is substance P? Substance P is a 11 amino acid peptide that is part of the tachykinin family. It has a slew of physiological effects, but mainly has been known for pain modulation and neurogenic inflammation. These effects are due to the binding and activation of neurokinin 1 receptors, which increase CAMP and DAG concentrations. What is interesting is that recent studies have suggested that substance P may exert some vasodilatory effects during cardiac events. Now, the mechanism through which substance P does this, not well understood. In fact, there is quite a bit of controversy surrounding it because it can also produce some adverse effects as well, such as cardiac remodeling. But this really just warrants further investigation, as there's potential for another preventative treatment modality to hopefully better the current state of cardiovascular disease. Because while there have been improvements in mortality rate, especially in the United States, cardiovascular health still remains a global issue. Preventable CVD events still arise due to poor management, and the cost still rises into the billions. So, the main research goal here is pretty much figuring out how substance P exerts its vasodilating and cardioprotective effects. So the procedure that will follow will consist of excising the left anterior descending coronary artery from fresh animal hearts, cut them into rings, and mount them on the myograph, which measures the contraction of these rings. From there, we pre-treat with a known vasoconstrictor like U46 and then introduce substance P to see if any relaxation occurs. We can also utilize NK1 antagonists, eNOS inhibitors, and others to pretty much narrow down the mechanism. So here are some of the results that we've gathered. And the effect, this is the effect that substance P has with a pre-treatment of aprepitant and NK1 antagonists. Now, as expected, substance P does not relax the artery in the presence of an NK1 antagonist, leading to the theory that this receptor might be involved in vasodilation. However, looking at the results with pre-treatment with L-name and nitric oxide synthase inhibitor, the effect is quite similar, suggesting that substance P has multiple mechanisms to vasodilation. And I would like to mention that while not shown on the slide, we treated the arteries with bradykinin after the experiment was over to test whether the endothelium was intact. In the controls of our experiments, we saw that some were damaged, and yet all of them still managed to vasodilate in the presence of substance P, which also suggests that it might be directly influencing calcium channels as well. So it's clear that there is still more that needs to be done, such as utilizing multiple antagonists in the experiments, examine other pathways such as prostaglandins or CAMP if they are influenced by substance P, and also looking to see if any eNOS expression is exhibited. But that is pretty much it. That's my works cited, and I'd like to take a minute to thank all of those who have helped me on this path, especially my PI, Dr. Ishfaq Bukhari, Dr. Han, Dr. Geyer, Dr. Campbell, the KaiCom lab staff, as well as KaiCom as well, for supporting me in this endeavor. Thank you very much.